tuberculosis and many other diseases. We're truly honored, sir, to have you here. It's great pleasure to again, so let's give a big round of applause and welcome, sir.
My grateful thanks to the organizers for having me here. It's a proud privilege to be back in BCP because the first time I tried to get into BCP, I was kicked out. That is, I didn't make the grade for PhD admissions here under Dr. Malla. And well, let's move on with the presentation. You see, I expected an audience that comprised equally of DPharm students as well as research students. So maybe my talk will be dumbed down a little for many of you with advanced understanding. This makes it easier. I can only touch the screen to manage it. So, with your permission, I speak on. Wander around here. With your permission, I'd like to speak on uncertainty. Yeah, I'm not on that road, Okay. So, we have talked about mathematizing our approach to understanding lots of pharmaceutical phenomena using ordinary differential equations and partial differential equations. When I say ODE, PD, I understand that a lot of us will get uncomfortable. This is an area that I don't really like, And moreover, you get bin longing. So if you have bin longing, all you have to do is to punch in the data and you get your model fitting and everything. So one doesn't really want to, very often, many students of pharmacy do not really want to engage with the modeling approach in terms of ODE and PD. But I was telling Priyanka ma'am that all of this approach to understanding pharmaceutics using ODE and PD is derived from chemical thermodynamics. Specifically on radioactive decay theory. When we say first order kinetics, we mean that the amount remaining will be proportional to the initial amount and that will happen forever afterwards, till everything is gone. Now, for concentration, theoretically, things will never run out. You'll get smaller and smaller and smaller and smaller and smaller and smaller halves of the pre-material electric time. And for most dosage forms, like tablets and capsules and so forth, it is probably necessary or sufficient to use ODE and PDE to predict absorption and distribution. Because, again, there is no denying the fact that the amount absorbed will be proportional to the amount initially taken in the GIT, and the rate of absorption will depend upon the concentration difference. Therefore, the rate will decrease as the amount of drug in the dosage form decreases. Delivered by the dosage form decreases. Things get slightly more difficult. So, when we're talking about deterministic processes, that is the rate of absorption of a drug from a oral dosage form like a tablet, a capsule, a syrup, something, we are looking at a deterministic process. And that's as deterministic as firing a cannon or a gun or something, because very simple equations govern the trajectory of the cannonball. In fact, in our school we used to write simple equations of parabola for this purpose, right? But as things get a little more complicated and you think of entirely predictable from the initial conditions. All intuitively know that most biological processes are smooth and cut it right. And I've chosen to illustrate the release of neurotransmitters, especially in the brain. You remember there's a synapse, and there are granules that contain hormones, and that degranulates and punches a lot of a bolus. A neuron punches a bolus of neurotransmitter into the bloodstream. When and how often and how much degranulation happens is stochastic. You can't really trace all the neurons in the brain all the time and say that, okay, this guy is going to fire this time, this guy is going to fire this much, and so on and so forth. So there is an unpredictability and uncertainty in the amount of hormone or neurotransmitter secreted by the neuron at any time. And this is crashingly important. You see, neurotransmitters are things that make you work in different ways in fractions per second, right? And so the time dimension gets extremely crucial when you're trying to model this, mathematize this, understand this to be able to predict. What I'm not showing you today is a bunch of work that I did starting from my PhD, but I think I will. Staying with the neurotransmitter thing, I remember, if you recall, the hypothalamus, pituitary, and gonad axis, endocrine axis. So the female hypothalamus and the male hypothalamus both secrete GnRH, gonadotropin release hormone. This has a special shunt blood vessel to take it directly to the pituitary. In the pituitary gonadotropes in the female secrete LH and FSH. In the male, they secrete LH and FSH. So far, there is no difference between males and females in terms of neuroendocrine control of reproduction. However, when LH find ovaries, they stimulate the production of progesterone and estrogens, right? And when they reach the testes, in the case of the male, they use production of testosterone. Now, these steroid hormones at the end of the cascade feed back upon both the pituitary and the hypothalamus to stop these neurons firing. And this is how you find that in the menstrual cycle, that is lasting typically 28 days in women of age, you find a rise, a peak, and a fall of estrogen and progesterone. In the fortunate male of the human species, hormonal changes that take 28 days in women are accomplished in 24 hours. Which is why young adults might find early morning erections as a phenomenon. But old fogies like me are not capable of anything because by now the pulsability in our hormone secretion has declined. So in the male, you see within 24 hours, you see rises. You see rises and falls, and correspondingly, you see testosterone coming up or down. Now, this is not a simple waveform, right? Lots of people have tried to model the menstrual cycle using the deterministic modeling approach, and they use differential equations and the roles of estrogen, progesterone, FSH in estrogen segregation was neglected. And there is compartment modeling. You are all familiar with compartment and non-compartment modeling. Okay. Having compressed ups and downs in LH and FSH and testosterone within 24 hours, we need fuzzy control. We really can't imagine a deterministic process saying that this person's neurons will fire so much GnRH at certain time by the clock. So we need fuzzy control, and the feedback control loop also contributes to the fact that there is uncertainty in how much GnRH will be released from which neurons at what time. So all of these controls require non-compartment modeling and stochasticity in terms of Brownian motion and entropy. Now, don't get scared of these terms. You know very well what is Brownian motion. right? I don't need to tell you that Brownian motion is zigzag random movement, and entropy is the tendency towards disorder. The trick is to constrain entropy and Brownian motion into handleable, let's say doable tasks. Otherwise, we let the time. Entropy means that we are not it. So that's very convenient way of modeling something. That is entirely not silly anything, but then doesn't work very much. I'm not going to discuss in detail these equations, but the reference is here, and both of these use a Hill function. Now, I don't think everybody is familiar with the Hill function. Hill function, names. Okay. Hill is an object like this, also the name of a man who sort of showed that decline is exponential mostly. You know what the exponential? Follow this kind of curve, but with constraints imposed by the deterministic first order model. So Hill function is a modified first order decay model with constraints to the rates being allowed for truly exponential decrease. No point discussing these equations because they are too complex and they are daunting in the sense that if I start saying this is when d by dt p LH is the possibility of LH, I lose your attention, right? And if you want to really use this equation, you will hunt it up and use it. So there is no point in my wasting your time and mine trying to explain to you what these equations mean. Suffice it to say that they are publication quality, they are well published, and they provide adequate modeling of hormonal processes of reproductive hormones secretion. We use MATLAB to program values and events equations to give us stochastic system. Equation of GNRS. This is six. So the graph on the left shows the secretion in terms of picogram per ml per hour, that's the rate, and this is the resultant concentration. This is pretty much like what happens in real young men. So that you have a peak at 5 a.m. and a decline by 20 the possible. And really fall off when you go to sleep around 24 hours. Why we are doing this is that in the 1990s, male contraception was a very big thing. There was no, let us say, pharmacological contraception for men. There was this thing called gossipol that the Chinese had been pushing, but it seems gossipol had very toxic effects on potassium metabolism, and they discontinued that. The top runner was long-acting esters of testosterone. Just as the female pill uses steroid hormones to suppress LH and FSH, GnRH actually. So also, if you notice from that diagram, that cartoon, testosterone feeds back upon the hypothalamus and the pituitary to stop the secretion of testosterone. And for spermatogenesis, just like the blood-brain barrier, there is a blood-testis barrier, so that in the testis, intratesticular concentrations are typically 40 times those of the peripheral concentrations. And remember, the peripheral concentrations are fluctuating very rapidly. But in order to maintain spermatogenesis, inside the testis, the amounts have to be 40 times than those that you find typically in the blood circulation. So, feedback control of GnRH secretion by long-acting esters of testosterone was a front runner, and WHO was doing people trials like crazy. The problem with those trials was that people who got the injections, they started losing hair, they started getting pimples, and what is most important is that as they release more of, as the testosterone plateaued, they had already inhibited the neurological aspects of hormone secretion. And these men felt terrible. They felt exhausted, they felt demotivated, and you know why? If you are using contraception, it stands to reason that you are having sex, right? If you can't have sex, then what's the point of this contraception? I mean, so that came to light. Now, we thought Oh, go false modesty. I thought that if we put in testosterone at this time, if we put in testosterone at this time, we should be able to stop this person. And consequently, if we are able to compute the next time the pulse is going to come, we should put in more testosterone just at that time. So the objective of computed modeling was to derive a parsimonious, that is the minimal possible up and down profile of testosterone that we could impose to suppress spermatogenesis but retain testosterone cyclicity, avoiding the side effects and retaining the motivation and performance of the recipient, which is what the good contraceptive is supposed to do. LH secretion, LH concentration, and this principle is something that we taught in school. Destructive interference. If a wave of testosterone meets a wave of GnRH, it was flat. When that happens, pulsatility is lost, spermatogenesis is lost, and performance is retained. So, this is the PK of transdermal patch of testosterone. After nicotine, testosterone, after scopolamine, the next thing to be tried was transdermal testosterone. So, this is the trial data. So, we get a peak of about 800 nanograms per deciliter, and the model that we find describes testosterone concentrations in this manner. So what we have to do is to find destructive interference, a waveform with which to stop this secretion. The method we used again was transdermal testosterone, but we used polymers, mainly PDP, PDAC, and natural rubber as anti-nucleating agents. If you remember your Thiel and McCabe engineering chapter on crystallization, you find that you remember that a crystal needs a nucleus to grow on. And if you have a polymer sitting around, if you have sufficient polymer sitting around, you can change the monoclinic habit of the testosterone crystals into crystalline dimensions in the nanometer range. And again now, dissolution of testosterone crystals is no longer a deterministic process. It becomes stochastic now. Depending on how much polymer you have, what reduction in particle size you will attain, release from the transdermal testosterone will be stochastic. And with combinations of polymers, we can have it make 1, 2, 3 peaks of this nature. After IV bolus, testosterone drops very rapidly. Short half-life of 15 minutes. If we apply in silico, the model is running, a stochastic differential equation model, giving you concentration of testosterone. Now you impose upon this, simple vector addition, you impose upon this another stochastic differential equation that represents the release of testosterone from these patches. And sure enough, you see that the magnitude as well as the frequency of pulses is reduced. More importantly, when you stop testosterone, that is, you pull off the patch, you stand it away. And this is exactly what we need. We don't want to sterilize the person, we just want them to have a reproductive holiday. So, at a greater magnification, you can see the same thing. And again, and importantly, you recover pulsatility both in terms of frequency and magnitude if you stop applying testosterone boundary. These are two rats which were sham operated, and these two received testosterone containing patches. You find that, remember the stages of spermatogenesis? I don't know if they make you do this anymore. We had to do stages of spermatogenesis. Stage 10, stage 12, and so on and so forth. So my student Ritu did not have trouble in identifying that after 7 weeks of application, after 7 days of application, you find arrest in spermatogenesis. And if you study the behavior of these male rats, there is one male housed with 3 females, the control had mating frequency of 0.78 at 8:30, 0.61 with 8.15 and 0.69. So there, even in rodents, clearly there is a variation in the frequency of mating without any intervention. And the deliveries per mating is also variable. 0.84 in the first 30 days, 0.66 here, 1 in the 71 to 18 days where the mating frequency is low. But highest number of deliveries per mating. In the sham animals, this is not changed much, but in treated animals, the ones that receive patches, 6 animals here, 6 animals here, 4 animals here, 2 here, because the other guys were sacrificed to look at their testes. So, and you find significant differences between mating frequency as well as deliveries. So that in the period where these animals were the most fertile, these animals did not sire any pups. We say, okay, this is a great thing to do. And I went after the life of many people, including Johnson & Johnson, that this is something that the world needs and they're looking forward to it. But by then I didn't know, I was young, I was idealistic. ये आदमी इतना हरामी होता है ना, males are simply not willing to shoulder the responsibility of contraception. They believe something horrible will happen to their testes. And I mean, it's a non-starter. When I went and spoke to their marketing people, they said, they don't buy it. I mean, you would beat them up when the Sarkar has to say, you have to wear this. Then it might be, but then that leads to another problem. If a man is wearing a transdermal patch and sharing a bed with a woman, the likelihood of the patch coming off and staying on the woman is high. And so we androgenize the female, which, again, is something not that Pharmaceutics wishes to do. However, mechanistically speaking, what happens if you make testosterone pulsatility flatten out? You find apoptosis in the pituitary gonadotropes, such that repeated feedback by testosterone is both a proliferation signal as well as a survival signal for these cells. And this was an important finding. So, tunnel labeling, you're aware? Tunnel, I don't think you're aware. Okay. When cells go into apoptosis, programmed cell death, their chromosomes have termini called telomeres. And these telomeres are enzymatically, let us say, controlled in terms of size by telomerase kind of enzymes. You use the tunnel is end labeling, say, EM is end labeling, terminal uninucleotide end labeling assay. What it means is that if you take a section of pituitary and you stain it with the fluorescent dye conjugated to tunnel, you find that cells that are apoptotic will light up. And you see massive apoptosis not much here in the control in the sham, but massive apoptosis in the pituitary, in the patch treated animals. Now we will switch gears and come to what Dr. Mala was kind enough to remember, which is another world first from our group. Again, no false much my brainchild. So, as you all know, tuberculosis treatment for drug sensitive TB takes 6 months. And 4 drugs in the initial phase for 2 months, isoniazid, rifampicin, pyrazinamide, ethambutol, and 2 drugs, isoniazid and rifampicin, in the continuation phase for 4 months. All of these drugs, anti-TB drugs, are toxic. I mean, it is just a trade-off, because all patients receiving these drugs, they report adverse effects, and it is really painful. Second, in terms of public health devastation, it is expensive. Because TB, I mean, in India, as in most of the world, TB drugs are administered as dots, directly observed therapy. So that a medical health worker has to go to the patient and observe the patient swallowing the drug. Why? Because these things are so toxic that patients have been known to put them in their mouths and spit it out. They would rather have TB than the side effects. we all are aware of the awareness level of our people in our country. So directly observation is a kind of policing. Now, I wish we could evolve such a program for transdermal testosterone for contraception, but I think it's going to be sociologically difficult. But anyway, if we were to, it's a WHO's stated objective that TB research is focused at shortening the regimen of treatment to limit toxicity and to reduce expense, public health expense. So we have particles, a dry powder inhalation, that can cure TB in mice and guinea pigs in 1 month. It is very, very safe for 90-day continuous daily dosing from up to 100 milligrams per day in 8 kg monkeys. So margins of safety are really high. And like good boys and girls, we use, we have installed capacity at CDRA. Only after my retirement, I don't think people in the department who are there have the guts to apply for approval Form 30 license, Form 29 license on Form 30 application. But things to bear about. We get 60% yield at lab scale. You see other people spray drying, you don't find people getting more than 40% yield. We get 60% yield. And lots of my colleagues all over the world are reporting particulate delivery systems with drug content of typically 10%. I laugh at them openly and say you are making excipient delivery systems with drug inside the bottle. And you're not going to deliver the excipient, right? Best formulation is one that contains no excipient. That's what the pharmaceutics people were taught. The best formulation has no excipient. But in order to formulate particles, we had to use a polymer and we used poly-L-lactide. This is different from PLGA, which is lactic co-glycolic acid. This is pure lactic acid and extremely hydrophobic and the highest molecular weight you could find. So that we intended to use this polymer more as a binder than a rate controlling matrix. However, getting two things as different as isoniazid, which is BCS class 1, highly water soluble, highly friable, and rifabutin. People ask why not rifampicin? We tried rifampicin first. We worked with lupin from 2000 to 2008 because they were interested in commercializing this. That's another story. But you guys pat me on the back and tell me that God, you must have worked very hard to get these three things in the same true solution without precipitating either isoniazid or the polymer or vice versa. How do we do it? We made a solvent system. And as pharmacists, I must share with you that the order of addition is key. You dissolve isoniazid in bisanol, and this is also an interesting story. We had our Sarkari labs at CDRA, and the ambient temperature around the time when I was working was something like 42 degrees Celsius. Does anybody remember the boiling point of methanol? 65. So it's hot methanol. When he went to lupin to transfer the technology, they had environment control. And the volumes of methanol they were trying to dissolve isoniazid, it just won't go. It might be very different 42 degrees. So I just took it between my hands and rubbed it together and lo and behold, isoniazid dissolved. And it dissolved rifabutin and PLA in dichlorophene. You start stirring this rapidly and dropwise add isoniazid in methanol so that precipitation does not happen. Now, once it is dispersed, it is made and you can spray dry it to solution. And for those interested in aerosol performance, the median mass aerodynamic diameter is 3.6 microns. It is said, you've all seen these figures. PM5 values are applied almost every day. So anything below 5 microns goes to the deep line. Above 5 microns, aerodynamic diameter, not physical diameter. Aerodynamic diameter is different from physical diameter in a very important sense. In the sense that it is dependent upon the shape of the particle and the relative density of the particle. So that old joke about what weighs more, a kilo of cotton or a kilo of iron. They weigh the same, but you drop the two from the luminar. Cotton will reach much later. And this is the aerodynamic diameter of the okay, so the relative density and so MMD is 3.6 microns. The geometric standard deviation between three batches is 1.4. The fine particle fraction less than 0.45 microns is nearly 80%, and stability will be at 3065 is 2 years. Now these are electron micrographs of lung sections of a mouse that had received inhalations by another apparatus that we invented, which is another story. But you see that this is a macrophage with a horseshoe lobe shaped nucleus that gives it, and 1, 2, 3, 4, 5, 6, 7, 8 particles inside its cytosol. These particles are inside their own phagosomes. Remember that when a mammalian cell swallows something, its cell membrane invaginates and the outer leaflet becomes the inner leaflet of the vacuole. And these phagosomes are rapidly acidified so that the protons pump into it, and they merge with lysosomes that push their enzymes into the phagosome. And here is a particle inside the phagosome that is interacting with the lysosome, and here is a lysosome that is actually pushing its contents into the phagosome. Now, one of the ways that Mycobacterium tuberculosis survives within these very cells, these are defense cells. The first arm of the innate immune system that are supposed to trap, swallow, and destroy all creatures, all particles that are depositing on the lung cell. But Mycobacterium tuberculosis has co-evolved strategies whereby it prevents phagosome acidification and phagosome lysosome fusion. And the bottom right picture shows you, if it is visible, a confocal micrograph in which the red part are particles that bear a red dye. The bacteria have been stained with green dye, and right here you can see a patch of yellow showing that these have co-localized. That is, this poor bacterium finds itself in the vicinity, close proximity of a dirty grade particle containing humongous amount of drugs. So no wonder it's killed rapidly. Intracellular targeting to kill the bacteria, and intracellular targeting to retain the drug in the lung space rather than into the systemic circulation. And here insulin, if you remember, was designed to rapidly go to the bloodstream. Here we wanted to retain. Why is the treatment of maintaining so long? Why 6 months? Because the granuloma of the tuberculosis patient are very poorly perfused, their blood vessels are dead, and the bacteria are living deep inside. So you need sustained large doses over 6 months to generate and maintain high concentration in the tubercular lung. These guys exactly do that. Now we come to the uncertainties. With that introduction. When you make particles by any scalable, industry scalable process, we get a log normal distribution of material. Some will be medium size, maximum size, some will be larger and smaller, and our particles happen to have this kind of log normal distribution of particle sizes. And we provided, as I said, a true solution. Now the excipient itself is a polymer, and what you get in terms of specification is the median molecular weight. So when you have polymer synthesis, you also get a log normal distribution of polymer chain size. So these two log normal distributions effectively turn each particle into a more or less unique building system. Unlike tablets which are trying to disintegrate, these guys are intended to retain their morphology for long enough to be taken up by macrophages, and since they are biodegradable but high molecular weight, they will retain their morphology for days and weeks, maintaining high drug concentrations inside macrophages where the Mycobacterium tuberculosis bacteria reside. However, drug release will be controlled as usual to the burst effect. Fick's first law. Anybody knows Fick's first law? Minus J is equal to D A C S upon D or H, which is right. Diffusion coefficient times the area times the concentration difference inside and outside, or this side and that side, upon the path length of diffusion. That will continue to apply. However, PLA does not swell and is not hydrated. Instead, it undergoes bulk erosion. That is, it falls apart like the cookie crumbles. It develops pores and channels that fill up, leach out the drug, and ultimately cause the thing to collapse into oligomeric particles, oligomeric chains. In 2007, we studied drug release used in online like boys and girls because regulatory pressures and peer review pressure, and we got publication in which we used the USP paddle, and to this we tie a dialysis bag containing the particles. As a control, we do not make particles but just mix the polymer and drugs in the same proportion that they are in the particles and study release. And we find that over 1 day, about 20% is released from the mixture, but at pH 7.4, less than 15% is released from polymer particles. It's even a little more in case of pH 5.2. With rifabutin, you have even less release, less than 5% in case of pH 5.2, and less than 2% in case of pH 7.4. So, burst release is minimum in the USP apparatus, and the observations that we have plotted are here. That is, black ones are ionized release at 7.4, and red ones are ionized release at 5.2. Greens are butyl at 7.4, and blue is butyl at 5.2. You see that the typical sigmoidal shape. And if you are fitting ordinary differential equations, you will not get it. ODE are designed, EDE are also designed to show you a smooth transition with rates changing depending upon the initial amount. This is not happening. And there's this very influential paper by Ekman and Salzman. It's called Mathematical Modeling in Drug Delivery, also by Bob Langer's group in Advanced Drug Delivery Reviews, that tell us that if you try to fit a sigmoid curve here, at first sight it looks like a sigmoid curve. If you just visually inspect these things, you know something has a lifetime that inflects and that type of curve. So a logistic equation is to be fitted there. But Salzman, Bob Langer, all the opinion leaders in this world, they are not happy with us doing what we call phenomenological fitting. Whereas Higuchi, Peppers, Kurzweil, Peppers, Weibel, and so on and so forth are mechanistic in the sense they have an explanation for each phase of the drug release. We had in 2007, we said, okay, this is closest to Kurzweil Peppers release, and that was it. But if we accept that there is stochasticity here, the stochasticity resides not in the mass of drug available in the release medium at any time, but the rate of release from particles. Are you able to make this distinction? When we study first order, let's say, release of drugs, we are looking at the mass, the weight, concentration actually, if I still see, the concentration change as a function of time. Here, we are looking at drug release as change in rates of release depending on size and molecular weight of the constituent particles. So the rate is the mass reaches infinity divided by an exponent of the rate of release at t minus t0 I. Right? And since we are not taught stochastic differential in pharmacy, it gets a little difficult. So here steps in AI. I don't know how to code Python. I ask ChatGPT, I ask Grok, I ask DeepSeek, and it's important to ask at least 3. Because individually, they hallucinate. They are capable of doing great things. I told it I want to fit my observed data points within one standard deviation. तो OD चल रहा है, बीच में interpolate करना है। क्या हो रहा है भाई? Then I looked at the script and I said, interpolate. And I said, no, don't interpolate, don't interpolate. Okay, it's possible to make them work for you, but only if you know what you are doing. If you try to use AI to do stuff that you don't know what, you are in very, very dangerous ground. Because these things are, especially the free versions, I am a confused man, I've said I didn't buy any one, so I don't know about the paid versions. But free versions are designed to please the user. It gives you what you want. So you want data fitting within one standard deviation, look, and hang the magnetic is not altogether. So I use the AI to write a script, use another AI to audit the script, and a third AI to critique it. What's wrong with this? I asked Grok once, give me EndNote ready citations. Manufacturer citation. But no, hey, you try PubMed, try Google, you won't find that. But it's so the pitfalls are out there. Be very careful, but then it is useful because it can do donkey's work. Which is why I am interested in not calling it artificial intelligence, but artificial stupidity and deceit. So, ASD is a nice term. Anyway, we induce stochasticity using standard deviation to the R square, and the time 0i at which release onset begins is a stochastic function, and then we make an ensemble. That is, each particle then becomes an individual drug releasing entity, and therefore ensemble average iris release is this way. Now remember, when we are studying drug release in an apparatus, we study for 15 hours. Now, it is rare that a physics lab will allow somebody to use a DNA with a apparatus of 15 hours. But okay, I fought my way and our students survived for 15 hours. But during the course of the study, at 37 degrees centigrade, both isoniazid and gluten experience degradation. So what you're seeing is an underestimate, because the amount released has been more, but since it is degraded now, it's lost to HPLC, so you don't see it. So you have to add back the true release in terms of ensemble plus in time and the degradation rate. Again, I'm not going to go through the, let's say, the tortuous parts of assembling this ensemble and not the actual way that entire stochastic is written. But in principle, you can do exactly the same thing to your application using Python, which is freeware, and free versions of AI, provided you know what you want. Don't ask it to do your PhD or MPharm project, PK, unless you have done it yourself the hard way. You can embellish it, but danger. Be very careful of AI. Right. So have you got this true release? The median is at pH 7.4 is black and 5.2 is orange, and the dotted lines are 95% confidence intervals. We are happier with the data points with standard deviations, but this is statistically, let us say, more satisfying in the sense we know the level of uncertainty is greater the larger the magnitude of release. So it may be higher or lower, but the magnitude of deviation from the modeling of predicted value is higher in proportion to the amount released. Similarly, for rifabutin, there is a little difference. As I said, rifabutin is BCS class 3, poorly water soluble but highly permeable. Here, we are putting in an exponential finer scale for the rest is almost the same, and a factor E which shows for slower dissolution of gluten in the release medium at least and pH bundles as well. And stochastically here we mean by tau I, which is a log normal lactic log of tau and sigma to the power 2. And correcting for true release, the same thing, exponential degradation rate. And we find that at pH 7.2 and 7.4, we can write at a release profile of different glucose. I said 15 hours for a digital apparatus is excessive for a shooting lab. And I also told you that the quantum of release is minuscule in 1 day. So we want to study for 15 days and we studied in vials. And we got very similar results in vials. Although vials are not stirred systems, the sink volume is much less, 900 ml here, 50 ml here. But we got plausible values for isoniazid at pH 5.2 and 7.4, and isobutyl at pH 7.4 and pH 5.2. Why is this important? Because the particles that I was talking about, I showed you earlier, if they deposit on the lung surface, macrophages immediately rush to capture them, phagocytosis. And when they phagocytose them, they start moving towards the granuloma because there is a gradient of chemokines radiating from the granuloma. You know what are chemokines? You know what are cytokines? Signaling molecules that say come here because we are calling you. So they come here. Now what this results in is what has been called in drug delivery the Trojan horse approach. That a cell is the drug delivery system. So the bacteria is sitting here and we are using a cell to transport the particle containing the drug to the periphery of the granuloma. And what this does is to create a sort of shell around the granuloma that is laden with drug. And any bacterium that wants to come out of this shell has to negotiate a minefield of drug molecules provided by drug release inside the macrophage. Now, when something is deposited on the lung surface, the first thing it will encounter is lung surfactant. Lung surfactant is composed of surfactant proteins and water, mucus can be viscous kind of substance, but it is enough to solubilize ibuprofen rapidly. And drug release from the particle on the epithelium of the lung, of the alveoli, and will have now two components. We will have some concentration in the epithelial lining fluid, which I call it ELF here, and we can calculate the volume of ELF in a mouse, and therefore the drug available for diffusion as 1.5 microgram per mouse per dose. And all of this mass is initially inside particles. As we saw, burst release is low. There's a lag time before it starts reflecting. So the time lag between phagocytosis and initiation of burst release, initial release, is fairly large, and we can also assume that this mark is initially inside. So how do we structure it stochastically? Log normal distribution of particle sizes, uptake per AM. Observing cultured macrophages, we find that if we expose them to a non-rate limiting number of particles, each macrophage is capable of taking up 1 to 5 particles. And the electron micrograph that I showed you had a macrophage in situ in the lungs with 6 particles. So we model uptake per discrete stochastic n from 1 to 2 to 3 to 4 to 5. So now each drug delivery system that is an alveolar macrophage may contain 1, 2, 3, 4 or 5 particles of any size and therefore released from the macrophage into the granuloma is stochastic. We also factored in phagocytosis time as an exponential lambda t and acidification lag of 1 hour. And what we then said was that before acidification, drug release will occur at pH 7.4, which is the physiological pH. But when it is inside the phagosome will be acidified, as we saw before using it lysosomes, and therefore release will now occur at pH 5.2. So just like in case of oral delivery for these two SGF, then we remove it and say SIF. We switch the pH here so that we see only loss in here. Now this is the problem. We are modeling only the loss in here because we say this mass initially inside, all the mass of the drug delivered to the lung is inside particles. But an analytical method, rather than a biomechanical method, lyses the particles and dissolves, and lyses the macrophages and dissolves the particles. So that sophistications of degradation before and after release, or rates of release from unspent particles, all of those are not incorporated in this model, but it's a very good enough estimate of loss only. At what rate will drug deplete from the macrophages? And we have the mathematical structure here. There is initial bolus for every macrophage. And step 2 is biphasic loss. And we have extended the release of isoniazic from 24 hours to 96 hours. A, because clinical PK is half-life is 40 to 96 hours. And it has two components: partially into lung surfactant, which is solubilization, and absorption into lung parenchyma, and a slow component binding to the epithelial lining and slow distribution from AM. It's a class 3 drug. And so we come up with a stochastic differential equation that incorporates all of these. In 2000, we published these concentrations, and we use the SGA ensemble to get these concentrations, which are quite similar to the ones that we find here, except that these are smooth slopes, not drops that we see because of lack of sampling in between. Again, the last step for today is doing the blood PK. Now, it is quite ironic that we are all agree that TB is a pulmonary TB is a lung disease. The bacteria is not present anywhere else, but our regulatory process and our peer review process wants us to report amounts in the blood. Now, the current challenge is, can we use the structures of and RNP release in cytosol to feed into systemic circulation? This will require again a different term from the parenchyma to the vasculation, and let us see what happens. So that's where I am at this time, and maybe when I publish, which I hope to do in this year itself, we will watch out for it and see how far I go. But the more important for us is the tissue peak. We find that inhalation gives you lots of drug in the lung, microgram 75, 125 microgram per gram of tissue, but very small amounts in the liver and kidney where drug toxicity is more highly manifested. The same for inhalation of rifabutin. Long persistent concentrations of rifabutin up to 96 hours, and little small amounts in the liver and the kidneys. Compared with the intravenous, of course, for absolute bioavailability, you want massive amounts in the, the largest amounts are in the kidney. In the case of isoniazid, and the largest amount again are in the kidneys and the liver in case of rifabutin. So, intravenous or oral anti-TB drugs are going to give you renal toxicity and septic toxicity, whereas inhalations do not, because they remain in the lungs. This is the PK in monkeys at 3 doses, and this is something that we have to model. Again, Win-Norlin gives us very nice publishable PK parameters as well as regulatory satisfaction. But when you do the experiment, and this is absolutely the last bit of stuff I will review, we infect animals at day 0, and in one case start treatment at day 10. Now this, I don't think I should go in much detail about it. Okay, we infected mice and guinea pigs with mycobacterium tuberculosis, and then we started giving them treatment, either with particles or with just the plain drugs formulated to the same particle size distribution. This is what happens. You have 12 mice, this is done at lupin, you have intravenous infection with 10 to the power 7 colony forming units and you dose for 3 weeks. This represents what is known as miliary taking. Lots of TB bacteria in the blood, also in the spleen and other organs, and an equal amount in the lungs. You find that inhalation alone, 80 microgram per mouse, that is 20 micrograms of isoniazid and soluble per mouse for 3 weeks reduces colony forming unit from 5 or 4.5 log to 0.5 log. And this is a log scale you agree? So 10 to the power 5 to less than 10. And if you combine half the oral dose with inhalation to take care of the extrapulmonary systemic circulated spleen resident bacteria, you find that in 3 weeks you clear the lungs and spleens of all bacteria. 3 weeks enough to cure. However, if you leave a cohort of these without treatment for the next 4 weeks, you find that inhalation alone starts to recrudescence. Whereas recrudescence, if you stop treatment in 3 weeks. So the minimal duration of treatment that we figured out was for 4 weeks. Again, combining oral dosing with inhalation dosing was sufficient to clear and keep the lungs and the spleens of bacteria after 4 weeks of no treatment. More kind of experiment. This is an aerosol infection instead of intravenous. That is, bacteria are localized in the lungs only. You find inhalation alone has cleared in 4 weeks. Oral prescription has no treatment for the next 4 weeks. You find that inhalation shows recrudescence, and combining oral with inhalation shows no bacteria in there. I think these are all third-party experiments, by the way. I didn't touch it. Neither did my students. They are Lucas people doing it. And they have no axe to grind about fudging and distorting and lying about. They actually wanted excuses to stop the work, but I can record this. So we went to ICMR and said, give us money for a clinical trial. They said, but then you are only giving 2 drugs by the oral route, whereas TB goes for 4 drugs. I said, yeah, government ठीक हो जाता है 4 तो आप ठीक हो जाएगा, that didn't work, so we had to do actual DOS drugs and human equivalent DOS doses. And again, very thankfully, we got 1 mouse lung with 1 colony forming unit. And I really thank this poor dead mouse, because this is one data point that shows that we are not manufacturing data. This is the one that got away, extremely proves the rule. And therefore, very, very happy that DOS plus inhalation discontinuation shows 1 mouse lung with 1 colony forming unit. And therefore, we maintain that inhalation plus oral dosing provides relapse-free cure in mice and guinea pigs in combination with oral dosing with just 2 drugs or all 4 drugs within a span of 4 weeks. And therefore, our trial is designed to simulation, again, no great tricks, just to show you that safety margins are very high. Very interestingly, the LD50 of human LD50 of highness is known. I wonder who did this experiment. Humans को खिलाते जाओ जबकि आधे मर रहा है. I think it might be Pfizer or Heck during the times of the Second World War, when they had a very large pool of experimental humans in concentration camps. Be that as it may, we have LD50 oral iris is 100 mg per kg, and safety margins are 16, 200, 15, 780, 10, in all cases almost 10, above 10. This work was initiated by Roli Sharma, who is now a professor at Kanpur University. Rifabutin was introduced by Roli did rifampicin. Someone who is now full professor at University of Albuquerque in New Mexico. Awadh, who is a full professor at University of Allahabad. Rahul, who is a scientist at Institute of Nanoscience and Technology, Mohali. Jatinder, who is at Takeda in the US. Amit Singh, who is at the National Jalma Institute for Leprosy. Anuradha, who's at NII, Mridul, who is internationally from NIMR, Atul is at USFDA, Sanket is at Torrent, Rajiv Ranjan has got multiple sclerosis and is back teaching in Bihar. Ashish Srivastava is in US. Reena Bharti is in the US doing a postdoc. Then Shiva Reddy is also on there. Asha is also on there. Sunil Raman recently got a job as a lecturer at Gorakhpur. And Trisha Roy is also doing a postdoc somewhere in the US. These are colleagues. It has been very well funded by CSIR, IQAC, DP, DST, Royal Society, Norwegian Health Council, Cultural Society. And that Dr. William Pharma Campus Pharma in the back. Oh, thank you for the attention. Why do we cannot throw? That's all we have. Yes, please. We are covering our bets that the simplistic model that is relying on ordinary differential equation does not mislead us as to the homogeneity of the problem. We are trying to capture the uncertainty that arises due to biological phenomena in and of themselves, added on to by variations in the dosage form.
In case of biological results, there are a lot of variations and
Yes, precisely. I mean, to me, it is not a question of taking care of biological variability. My concern is to account for it. Mathematically, mechanistically, how do we account for why biological responses in pharmacology, in immunology, in endocrinology, all over the place, they vary so much, as in humans. You see, in human pathology, fixing a dose has always been a problem. Body surface area, weight, gender, age, pediatric formulations, so on and so forth. So, what we are trying to do is to account for biological variation. When we say dose is this much, so on and so forth. Now, ideally, in an ideal world of personalized medicine, each patient should get a dose. Look, I told you, I write my scripts very well. The prerequisite is to be able to appreciate the sources of stochasticity in observed phenomena and also be able to distinguish between what is sufficient to explain a chemical thermodynamics approach to study decay or whatever, some process through ODE or PD, and to realize the limitations of ODE in PD by flattening out lots of things over very large space. So, I give you two examples. One from endocrinology, where you look at stochasticity as arising from degeneration of neurons, and stochasticity in drug delivery occurring because of variations in discontinuous variations in particle size of testosterone, and also habit from crystalline to amorphous. And to make an amorphous stable formulation is also not a joke, but fine. So if you want to use stochastic differential equations, don't set out with the idea that I want to do it deeper. You look at your process first. Does it admit to variation because of stochasticity? Or is it deterministic enough for us to use ODE and PD? If it is deterministic enough, like suppose we are making an oral dose form, which is a very noble pursuit that all of us should do. I made tablets for 1 year in Bombay itself for health pharma, and my formulation is LED tablets that is still being sold in the last 10 years. The Comptil tablets. Comptil is prochlorperazine. Anyway, let us not take cannon to kill a mosquito, right? If there are simple approaches that are acceptable to reviewers as well as regulators, you should definitely prefer them. You go to Bayesian or stochastic modeling only if there is something dissatisfying about ODE and PD being able to fit complex phenomena such as neuron derivation, Lafarge effect, transfer from epithelial lining to granuloma, and transfer from these cells into systemic circulation. I mean, you intuitively can think that these will be stochastic. They're not going to be different. It can happen in any time, in any magnitude. If you have something that of that nature, that you can't predict what will happen in the next time, you will see. And you at least 3 AI agents, 3 ones every year, and never believe what they say unless you check it yourself.
Can we apply this in pharmacokinetics?
Sure, I will do that exactly for the evolutionary. I showed you that I've got tissue pharmacokinetics in terms of cellular level pharmacokinetics in macrophages. Now the next step is to go from parenchyma, from macrophages to parenchyma, from parenchyma to systemic circulation, and to secretion and excretion. Now that we have it in hand, I can probably finish in a couple of days. Only I'm retired, so it's very difficult to make me work.
So what do you get actually at the end? You get a range of some data.
You get a predicted profile.
Predicted profile, which is not the accurate with this one.
No.
That is what you get it from OD and AD.
There are statistical measures. One is skewness and kurtosis. Since these are ensembles, we look at the skewness and kurtosis. If they are within the acceptable ranges, then the kurtosis is not going up and down here and there everywhere. There is a smooth skewness and kurtosis. Then that's it. The other is the approach that increasingly PK is using. Things like IQAC information criteria, AIC. Do we minimize AIC by fitting parameters more closely using ST as opposed to OD and PK? That is another approach here. Because you see, there are some tragedies. In case of drug release, we reported observed values. But however objective they are not telling us how much drug was degraded in the interval that is happened. So that has to be coming by calculation. So, and when drug release is stochastic, drug degradation is also stochastic. Error is always multiplied.
So what are the tools we can use after generating the script? What are the free things available for this?
Python. Download Python. I have 13.3. I think I will generate it soon. And run the command prompt. And with Windows, type Python. If you run as administrator, it's important to run as administrator because otherwise it won't write. Then when you got it there, you go to the command prompt, have a script ready, and say Python script.py. Join this. You want to demonstrate this? Huh? When you have time, your phone is here. I kept it there because it's often produces interference 15, 17 minutes. 15 minutes super short. More? From Supersede? ये तो नहीं पूछ रहा है कि बोर कर दिया कि कह रहे हैं चैट की बात करो खुद तो कर रहा है फिर कह रहा बात करो No, but I'm serious about it. You need 30 years of experience to be able to have an insight that will not be hoodwinked and taken in by AI. क्योंकि झूठ बोलता है, साफ साफ झूठ बोलता है, शॉर्टकट लेता है, बदतमीजी करता है, हर तरह का जो अनपेड लेबर जो करता है ना, सब लेकिन नहीं है स्टूडेंट सजेस्ट करो यार तो पाइथन कोड कर दो तो स्टूडेंट करते हैं मैं बुढ़ापे में नहीं सीख पा रहा हूँ बस ट्राई करिए लिखने से क्या है तो आता नहीं है यू लूज द स्किल ऑफ रिकॉलिंग एंड रिमेंबरिंग एवरीथिंग मेरा इनसाइट लेवल तक प्रोसेस आ रहा है जहाँ पर
Thank you.